Welcome, curious minds, to another deep dive. Before we plunge into today's fascinating topic, just a quick heads up. This deep dive was prepared with the assistance of AI technology. However, all scientific content has been thoroughly reviewed by cannabis genomics researchers to ensure accuracy and integrity. All right, let's unlock a truly ancient story today. We're talking about cannabis sativa. I mean, this plant's history with us goes back, well, millennia. Think about it. Archaeological evidence suggests domestication in East Asia, maybe 8,000, maybe even 12,000 years ago.
Yeah, that's incredible. Right up there with wheat and maize, an old relationship.
Exactly. And for ages, it was this multi-purpose powerhouse, wasn't it?
Yeah.
You got fiber, food from the seeds, and, well, obviously the medicinal and psychoactive uses. It spread everywhere, eventually splitting into the hemp types and the drug types we recognize.
It really is remarkable. It's seen these cycles, like you said, cultivation, consumption, and then crackdown. But the big thing, the real picker, was modern prohibition. Especially in the 20th century, it basically slammed the brakes on cannabis genetics. Just put it in a scientific deep freeze.
Maybe deep freeze, yeah.
For decades, breeding, germplasm development, severely restricted. It's almost like it got stuck, scientifically speaking.
That's a great way to put it. So now we're essentially playing catch-up, right? Trying to undo decades of lost time, bring cannabis genetics into the modern era like other crops.
Precisely.
Because things are changing. Recent laws, like the U.S. Farm Bills in 2014, 2018, they've really opened the floodgates for hemp research and production.
A huge shift.
And it's not just about the traditional drug market anymore. It feels like we're unlocking potential for new medicines, sustainable materials, all sorts of things.
Absolutely. New applications are emerging constantly.
But even with this new freedom, there was still a major hurdle, wasn't there, for really advancing the genetics?
Oh, definitely. Historically, trying to piece together the cannabis genome,
Yeah.
it was a mess.
Like that ripped treasure map analogy you mentioned earlier.
Exactly like that. Previous genome assemblies were inconsistent, riddled with gaps, poorly aligned. You couldn't get accurate gene annotations, just loads of confusion. It slowed everything down.
You can't build the blueprints wrong.
You got it.
Yeah.
Which brings us to the solution, the core of what we're digging into today, the absolute need for better, more diverse, more accurate genome data.
And the breakthrough.
The breakthrough is the complete, really high-quality genome sequencing of a specific cannabis sativa cultivar called pink pepper. This wasn't just a small step, it was a massive leap.
And not just any old sequencing effort, I gather. It sounds technically quite advanced.
Oh, it was. Really impressive. They used this three-way approach. Combined PacBio SMRT sequencing, Illumina, and Oxford Nanopore. Basically the best high-precision tools available.
Throwing the kitchen sink at it, technologically speaking.
Pretty much to get the clearest possible picture. And the result, well, think about those old fragmented genomes with hundreds, maybe thousands of pieces. The pink pepper assembly, it brought that down to just 17 large, well-connected scaffolds. It's incredibly continuous, incredibly organized.
Wow, 17 compared to thousands.
Exactly. And it hit the highest completeness scores, these things called BSGO scores, among all the cannabis genomes reported so far. It really set a new standard.
Okay, so we have this gold standard reference genome now with pink pepper. What does that immediately let us do? What doors does it open right away?
Huge doors. For you listening, this is key. It's the foundation for building what's called a pangenome. We'll get into that.
Okay.
It turbocharges cannabis genetics research, breeding programs.
Yeah.
It refines all the genomic tools we use, whether we're aiming for specific cannabinoid levels or discovering entirely new traits. It's the map we needed.
Got it, the foundational map. So let's dive into that core idea then. Pangenomics. Sounds big. What exactly is a pangenome?
Yeah, it sounds complex, but the idea is pretty straightforward. Think of it as the complete genetic library for an entire species. Not just one reference book, but all the genetic information across all its varieties. The whole collection.
The whole collection. It includes core genes, the ones every single variety has, the basics. Then accessory genes found in some, but not all, like optional chapters. And finally, unique genes. The really rare stuff found in maybe just one variety.
Okay, so it captures all the shared stuff and all the differences.
Exactly. The entire genetic mosaic.
And how did researchers actually build this for cannabis, given its complicated past?
Well, it was a huge undertaking. They constructed this cannabis pangenome using a massive amount of data. Get this, 181 new genomes plus 12 older ones.
Wow.
From 144 different plants, males and females included. It gave them this incredibly broad and deep view of the species' genetic landscape. like comparing thousands of family photos to understand the whole extended family tree.
And what's really fascinating here is that this revealed just incredible diversity, right?
Yeah.
Surprising levels of genetic and structural variation for just one species.
It really was surprising, especially after decades of that genetic deep freeze. When they broke it down, only about 23% of genes were core, found everywhere.
Only 23%. Yeah. About 55% were nearly core, so in most, but not all. Then 21% shell, less common. And tiny fractions were cloud or unique. It really underscores the structural variation.
So lots of differences between varieties.
Loads. Although, interestingly, those core genes, they're often involved in things like terpene production and defense responses, really fundamental stuff for the plant.
Okay, but despite this massive effort, it sounds like we're not done yet. There's unfinished business.
Definitely. The data shows that even as you add more and more genomes to this pangenome, you keep finding new, unique genetic sequences, these things called CARMers.
So the library is still growing.
It is. It strongly suggests there are still significant chunks of cannabis genetic diversity out there globally that we just haven't sampled yet. Huge potential for discovery remains.
Can you give an example of where that potential might lie?
Sure. Certain Asian hemp varieties, like one called Juvier H22-1061002 or another named Yunma, they consistently show way more unique CARMers. We're talking over 10 million sometimes. Compared to, say, varieties from well-established breeding programs, like some Oregon CBD lines, which might have less than 1 million unique CARMers. It clearly points to areas, particularly in Asia, as potential treasure troves of untapped genetic diversity.
Fascinating. Okay, let's zoom in a bit now from the whole library down to these things called haplotypes. What are they and why should we care?
Right. So think of a haplotype as a specific set of genetic variations or alleles located close together on a chromosome that tend to be inherited together as a single block or unit.
Like a prepackaged set of genes.
Kind of like that, yeah. A genetic recipe block you get from one parent, and they're super useful because they act like barcodes. Researchers can use them to track traits like specific cannabinoid profiles or disease resistance through generations.
And these new, super detailed haplotype resolved assemblies, what do they let us see that we couldn't before?
They provide this incredibly comprehensive framework. We can now clearly see these things called aligned runs of homozygosity, or AOH.
The AOH, a bit of a mouthful.
It is. But think of it like finding long stretches of DNA text that are absolutely identical between the copy inherited from the mother and the copy from the father.
Okay, identical paragraphs in both parent books.
Exactly. And if a plant has lots of these long identical stretches, these AROHs, it's a strong signal that its parents were closely related. It indicates inbreeding.
Ah, okay. So it's a measure of genetic relatedness or diversity within an individual plant?
Precisely. Fewer ARHs mean more genetic variation, more heterozygosity.
And here's where it gets really interesting. What do these ARH patterns actually show us in real cannabis varieties?
Well, we see clear differences. For instance, cultivars like FL58 or GRV1, they show really extensive ARH, long stretches of identical DNA confirming high levels of inbreeding.
Okay, so quite homogeneous genetically.
Right. But then you look at some of those Asian hemp samples we mentioned, like GVA H22-1061002 or the Compulti cultivar from Europe.
Yeah.
They show the opposite. High heterozygosity, meaning very few short ARHs, and they often have those large numbers of unique comers we talked about.
So diverse parents, lots of unique genes.
Exactly. It paints a clear picture of their rich genetic background, contrasting sharply with the highly bred lines. You can almost see the history of breeding choices written in their DNA.
Amazing. So pulling this all together, the pangenome library, the haplotype blocks, the AROH insights, what are the practical real-world applications? How does this change things for growers, for medicine, for industry?
It's fundamentally changing selective breeding and how we develop new varieties. With these detailed insights, breeders can now pinpoint the specific genetic variants linked to desirable traits.
Like mapping genes for yield or resilience.
Exactly. Higher cannabinoid yield, better resistance to drought or pests. You name it. It allows for much more targeted, efficient breeding, almost like designing plants with specific features using a genetic toolkit.
Now, you mentioned something intriguing earlier, this paradox with the main cannabinoid synthase genes, THC, the VHES, CBDS. They're in variable regions but show low diversity themselves. What's going on there?
Yeah, that's a key finding. It strongly suggests these specific genes faced intense selective pressure during domestication. Humans basically picked the winners, the most efficient versions for making THC or CBD over and over again.
So we narrowed the options down drastically.
We did. It led to a very limited set of functional versions, often linked to specific genetic structures nearby. So while the neighborhood, genetically speaking, is diverse, the cannabinoid factories themselves are pretty standardized in many lines. It actually makes breeding for novel major cannabinoids quite challenging.
Okay, so selection was strong there. But how does that contrast with other traits? Where do we see lots of diversity to work with?
Oh, plenty of other areas. Genes involved in fatty acid metabolism, think seed oils, overall plant growth, defense responses, and especially terpene biosynthesis.
The aroma and flavor compounds.
Right. Those show huge diversity, lots of variation in gene copies and versions across the pangenome. It means there's massive untapped potential for developing strains with unique smells, tastes, or maybe even new oil profiles beyond just focusing on THC or CBD.
And speaking of cannabinoids beyond THC and CBD, what about the rare ones? THCV, CBDV.
Yeah.
What are this genomic deep dive revealed about making those?
Significant progress there, too. Research pinpointed a cluster of genes on chromosome-7 acylipid thioesterases, ALT3 and ALT4, strongly linked to making these rarer three-carbon side-chain cannabinoids like THCV and CBDV.
Okay.
And it gets more specific. There's another key piece, a tiny 2-base pair deletion in a gene called BKR on chromosome 4. This deletion seems crucial for producing these varying cannabinoids efficiently.
Wow, a tiny change with a big effect.
Absolutely. And interestingly, tracing these genes suggests an Asian origin for the varying cannabinoid trait used in some breeding projects. We can follow the genetic breadcrumbs back through history.
Incredible precision.
Yeah.
What about traits like flowering time? We know cannabis sex chromosomes are ancient. How has human selection played into when plants flower or adapt?
It's a fascinating evolutionary dance. Take the Gigantea gene GI. It's a core part of the plant's internal clock regulating flowering. Well, this GI gene shows really significant genetic differences between typical marijuana populations and hemp populations. It's a clear molecular signature of how humans have selected for different flowering times, depending on whether they wanted fiber or flowers.
We've literally shaped its internal clock over time.
We have, and there's more. Remember that super gene idea?
Yeah, on chromosome 1.
That's the one. There's a large inverted section of DNA there that seems to act as a single unit. It contains a gene called PRR3, which is strongly linked to the autoflower trait.
The trait where plants flower based on age, not daylight hours.
Exactly. It's like an internal timer overrides the need for changing day length. A total game changer for cultivation, allowing multiple harvests or growing in regions with unusual light cycles. And now we have a specific genetic target for breeding that trait.
So beyond understanding the genes, how does this help improve the tools for breeding? Are we getting better at, say, finding the right genes without accidentally dragging along unwanted ones?
Yes, definitely. One major improvement is tackling reference bias. This is where your DNA reads might stick better to the genome map they most resemble, which can skew results.
Okay.
High-quality pangenomes act as a more neutral, comprehensive reference. They reduce that bias, giving a truer picture of any given plant's genetics.
Can you give an example of overcoming that?
Sure. Mapping the early one flowering time locus. Using older fragmented references, researchers got a huge fuzzy location, maybe 40 million base pairs wide, like searching a whole neighborhood.
Not very precise.
Not at all. But using newer assemblies and that pangenome consensus approach, they narrowed early one down to a much tighter region, around 2 million base pairs. Much more like finding the specific street address. That boosts mapping precision enormously.
Makes breeding much more targeted.
Yeah.
What about how genes get shuffled during reproduction? Understanding recombination.
Crucial for breeding. Cannabis shows this strong pattern called periphery-biased recombination. Most of the genetic mixing happens near the ends of the chromosomes.
Okay, lots of shuffling at the tips.
Right. But the middle sections, much less recombination, they're kind of cold spots.
Yeah.
This matters because of linkage drag.
A linkage drag.
Yeah. It means a good gene you want might be physically stuck close to a bad gene you don't want in one of those low recombination zones. It's hard to break them apart through breeding. Understanding this landscape helps breeders devise smarter crossing strategies to try and separate the good from the bad.
So, with all this potential untapped diversity, precise tools, what's the big picture for the future? What potential are we still sitting on?
Well, the variation in those fatty acid genes like ALT and BKR is really exciting. It hints that cannabis might have huge untapped potential for its lipid metabolism. We could potentially breed for novel seed oils, maybe even biofuel.
Beyond cannabinoids entirely.
Exactly. Which really underscores the need to look beyond the usual suspects. Hybridizing with diverse lines, especially those outside the common Northern European hemp pool, could unlock completely new traits and chemistries.
Which brings up an important point.
It really does. The conservation and active use of diverse germplasm, particularly from places like Asia and including wild relatives, that's going to be absolutely critical. If we want to realize the full potential of cannabis, whether for medicine, industry, or agriculture, we need to embrace and explore its entire genetic heritage. We've only just started.
That brings us to the end of today's Deep Dives. We explored the foundational role of the pink pepper reference genome and how pangenomic and haplotypic insights are really shaping the future of cannabis breeding and research. Thank you so much for joining us. We hope you found it informative and engaging. Stay tuned for the next Deep Dive, where we'll explore how these genomic technologies are continuing to transform cannabis science and maybe even impact agricultural practices far beyond just this one fascinating plant. Until then, stay curious, stay informed, and thank you for listening.